Gene editing is undoubtedly one of the most exciting fields in modern research. The ability to fine-tune an individual's genetics to cure diseases and improve lives is an exhilarating notion for any biologist. For many years, we've been testing our genetic manipulation techniques on crops, and we have developed transgenic plants that have been engineered for improved nutrition. For example, researchers have developed a strain of rice called golden rice that contains genes that encode the enzymes to synthesize a precursor of vitamin A. This has helped to alleviate vitamin A deficiencies, which affect roughly 400 million people and has the potential to cause blindness if extreme, such as in third world countries. Additionally, this golden rice also contains genes for a protein that functions in iron storage. This ameliorates iron deficiencies, which affects roughly one-third of the entire world population. More recently, though, we have moved on to deciphering how to safely genetically modify human cells and eventually, hopefully, people. At the forefront of this research, the disease sickle cell anemia has been the main focus of many of the experiments done with genetically editing human cells. In the past, researchers have performed gene editing experiments in an attempt to treat sickle cell anemia by using viral vectors that are capable of inserting DNA into the host genome. The major problem with using viral vectors is that after use, you have to perform an analysis to identify at what places in the genome the DNA was inserted. So essentially, this method is not very targeted to a specific site, and it ends up causing issues within other areas of the genome. This issue of a lack of specificity was solved by the discovery of the CRISPR-Cas9 system. According to Corey Smith in a research article from 2014, after numerous gene editing procedures were performed in a single organism and its entire genome was sequenced, they found that there was not a single off-target mutation in the entire genome by the CRISPR-Cas9 system. This shows how remarkably specific and promising this technology is. The CRISPR-Cas9 system is composed of a protein called Cas9 and a segment of RNA that guides the Cas9 system to specific sequences of DNA to cut it. This system is part of the bacterial immune system, in which the Cas9 system uses the RNA to recognize foreign viral DNA and cut it before it can cause issues. The Cas9 protein makes a double-strand break at its specific DNA site, which, when done in human cells, the cell attempts to fix it, but that actually results in random nucleotides being inserted, and that inactivates the gene. The Cas9 protein can also be modified to give it other functions. One of the most exciting of these is to couple the Cas9 protein with another protein capable of changing the nitrogenous base present at a specific site. This is uniquely exciting for sickle cell anemia, which is caused by a single point mutation, and thus we can restore it with a single point restoration, if you will. Many methods have been designed to deliver the Cas9 system to cells, but one of the most common is called lipofection, which uses a lipid structure to deliver the system to cells. Bilayered vesicles called liposomes can be formed around the Cas9 complex, which are then either endocytosed or those can simply merge with the target cell. And then that way it administers the complex. A novel delivery method that has been proposed is using a metal inorganic palladium cage, as shown on the right. So evidently there are very, very many delivery methods that vary greatly in their efficiency and their expense, which both need to be assessed when selecting a delivery method. Most other research has been performed to knock out the gene responsible for sickle cell anemia, but for this project, I plan to assess the true capabilities of the Cas9 system by investigating the feasibility of restoring the single nucleotide responsible for the mutant gene using a modified Cas9 system targeted to that specific residue. This will include investigating modified Cas9 systems capable of the base transformation and investigating the efficiency and expense of different delivery methods for the Cas9 complex.